Polymerase chain reaction is a process of in vitro DNA replication for amplification of a specific DNA sequence. It's a method developed by Carey Mullis in 1985, and this method uses the principle of heating the DNA strand in an automated thermal cycler machine. So here it will be using small microtubes which are responsible for carrying out the process. You can see here in the picture, and all these microtubes are used for the process entirely, and we can find they have the buffer loaded in them. The buffer can be found preloaded in the like microtubes. They are already purchased like that. So the entire setup ultimately will have a large number of this nucleotides triphosphates, which are deoxyribonucleotide triphosphates. Then we have this the buffer loaded in the microtubes. The fragment of DNA that is going to be amplified, that DNA fragment will be loaded in it, and along with it there will be primers or small strands which have a complementary sequence with that of the specific gene preceding it and following it. We'll come, well, the expression will be done so in that time, and right there it will be all loaded into that same microtubes, and along with it will come a special enzyme called the thermus aquaticus DNA polymerase, or in short TAC polymerase. It's actually a thermostable DNA polymerase isolated from specific bacteria which grow in the um, Yellowstone National Park or any kind of volcanic hot geysers. So in that places they are found, and there we can see this bacteria growing there, the Thermus aquaticus, which has the normal or optimum range of growth in the like around 70 degrees Celsius, so 6 to 70 degrees Celsius. Now in that range, so this bacteria, if it is able to survive there, that means all its enzymes can also be able to survive there, resulting in this particular DNA polymerase, the TAC polymerase, being able to survive in that temperature range, which helps the scientist to synthesize the DNA in vitro without any contamination. It is all achieved by this automated or programmed thermal cycler machine, in which the microtubes are put carrying the Taq polymerase, the DNA sample, the primers, the deoxyribonucleotides, everything loaded inside, and then by the program this machine changes the temperature in which everything is done. So the temperature is kept high first of all to achieve the denaturation or opening of the DNA strand, followed by the temperature is brought back down to initiate the annealing process. Then it is raised again to achieve the extension of the or the working temperature of the polymerase. This all can be programmed such a way that the machine can run the cycle for a single short PCS cycle where everything is kept short, or it can be kept for a long cycle in which the extension is carried out for a longer period of time. Now, when the extension is being carried out during that time, the DNA sample is first of all the temperature is increased to 95 degrees Celsius, which will break open all the hydrogen bonds between the DNA strand, and it will become two single strands. Now then the annealing is being achieved by bringing the temperature back down to 55 degrees Celsius, in which the primers that are generated, its preceding and following sequence. So these two sequences, they are being noticed and observed, and then primers are generated for both the strands, and these primers will come and bind to it. And after binding of the primer, now it actually gives the 3' hydroxyl end for the DNA polymerase to extend, because every DNA polymerase works the same way. Now when that is achieved, that very moment the primer, the temperature of the entire system is again brought back to around 72 degrees Celsius, in which the process of extension will take place. Now during the process of extension, the DNA polymerase works in that particular range of temperature, and it is desired to work in that particular temperature, as I mentioned, to avoid contamination. The contamination can be of the DNase enzyme or any other enzyme which may interfere in between the process, and by maintaining a very high range of temperature, those all contamination can be avoided, because all other enzymes, they normally work in a temperature range of 37-40 degrees Celsius, whereas the normal optimum range of working for this DNA polymerase is 70. So keeping a high temperature will prevent other enzymes from working. So now the tag DNA polymerase can extend the complementary strand by simply polymerizing the deoxyribonucleotides which are already present in the buffer, and then it will form the strand, the complementary strands, the double strands, and that will create two new DNA strands now. Now both this DNA strand can again act as a template for the next cycle. So it can be repeated as many times as desired. And every time the same thing is going to happen. The temperature will be first increased, then brought back to for the annealing or the joining of the primer, then it will be brought back up for the extension, and then finally the cycle will be repeated. Each and every of the cycle will actually create multiple copies. As you can see, it is 2 to the power n plus 1, the n being the number of cycle. So every cycle we have numerous number of copies of DNA being formed, ultimately helping us in amplifying the fragment of DNA. So this amplification can help us in detection of pathogen. We can increase the number of viral or whatever pathogen DNA that is there and can identify them. Diagnosis of a specific mutation that can be present in the cell, or we can go for the DNA fingerprinting. Not only that, it can also helps in the study of paleontology, where we can like mummified human bodies or any other fossils or anything, if anywhere, not fossils, not like that, but anywhere if we find a small DNA fragment, we can extend it and ultimately can determine the features and properties of that particular individual which was there previously. So these all are the applications of ultimately the process of PCR.